candidate for the Health Equities Initiative Professor of Biology. And she's giving her research seminar today entitled, what's on the first slide, so I'm not going to even, I tried this, Inner-Ear Organogenesis, and this next part, Sensorineural Regeneration. So the other things I can say very well. Dr. Serrano received her BA with distinction from the University of Rochester in physics and her chemistry minor. She got her PhD at Stanford in biophysics with Peter Getting, looking at electrical signaling in neural cells. And she did a couple postdocs, one with Robert Shimke and one with Susuma Hagiwara, who apparently is an amazing neurobiologist, cell biologist. Basically, one of the things we're looking for in this talk is someone who's a strong researcher, who has had significant grantsmanship, who has a significant leadership experience and mentorship experience. And I can go on and on about how this particular candidate has met all of those criteria and more. But I want to give her the time to really talk to us about her research and to tell us the various ways that she's looked at this wonderful organ. Is it an organ? Okay.
It's lots of little organs, but you'll know that within 20 minutes.
Anything below cells, I got everything above cells.
It's really wonderful to be here addressing this audience. I feel very honored to have been invited to interview for this position, and especially because I see in the audience people whose careers and mine have overlapped, and for whom I have so much professional admiration for their achievements as scholars and as colleagues in the larger crazy world of academia that we all belong to. So I have a very large task today, and that is to present an overview of the research in my laboratory. And we've been working hard, so there's a lot of slides. And the purpose of this is for you to get to know me, know a little bit about how I think, and what we've done, and even more so, where I see my research going, because my research right now is very exciting to me, and I'm very excited about the possibilities that I see here for my research to continue, in addition to very intriguing opportunities by this unusual position, the health equities position. So today what I'm going to do is briefly introduce the system that I've worked on, the inner ear and the mechanosensory hair cells. In my experience, very few people know this system in detail. I certainly didn't know it when I started working on it. I came to this field as an ion channel biophysicist, and it has been a wonderful journey. So I'm going to introduce the cells very quickly, mechanical structure in general, and then I'm going to provide an overview of the motivation for our research and highlight some of our accomplishments. And depending on the time, because I do want to leave time for discussion, I may move through those slides, but I know I'll be meeting with people later. We can pull out the computer and go through them. And then what I want to talk about is where my research is going. And I'll talk a bit about activities I've been participating in the last year. I had a chance to be part of an MIT Systems Roadmap initiative last year, and I'll be talking about that. And just really, and as I start talking about that, I'll talk about how I see the face of science changing and opportunities that challenges that all of us are facing provide. And so I'll be talking about research, and so I'll be addressing the areas that I am interested in, new areas, systems analysis. Our research is moving towards regeneration, toxicological work. And then this is just the surprise of being in academia and meeting cool people. That suddenly I'm doing a lot more in an area I never thought I would be, nanoscience and bioimaging. So for those of you who are students, it's to say that if you're self-motivated and inquisitive, you'll never be bored if you are a scientist in a university. So I'd like to begin first by talking about mechanoreception and the senses of hearing and balance. And I think of mechanoreception as something very broad. It's really an ancient sense. And the reason is that organisms must sense pressure forces. All of us are earthlings. And what that means is every organism on the planet must be capable of sensing the gravity vector. And so cells and animals have particular structures that let them do this. And that's why I love to show a mangrove and fabulous roots, because that's an example of how gravity reception is so essential that plant roots actually need to develop and direct their growth in regard to that. And so, but pressure forces, so I'm a physicist, so I'm going to present this in ways that are integrated. So what are some of the pressure forces? Gravitational field, contacts with surfaces and fluids. If you work on the heart, you're very aware of that. Osmotic gradients can create pressure forces. And of course, a system near and dear to me, the sense of sound and the sense of audition. And another sense that everybody here is pretty much sitting upright, so you have a sense of proprioception, the ability to orient and sense your body in space and to maintain balance. And so mechanoreception is receiving more interest, and it may be very important in tissue and organ engineering for those of you interested in that. So mechanotransduction is a mystery at the molecular scale compared with what we know about light and chemical detection. This is just to remind you that mechanoreception is more than the ear, but that in fact that all organisms have specialized cells distributed through the body for detecting pressure. Okay, so moving to the inner ear. The inner ear is the organ of force reception. I see this as a large force sensor. It is the organ of force reception for the senses of hearing and balance. And part of the way I view this sense now, although I work as a biophysicist at the cellular level, I've come to appreciate what this does for the existence of the organism. So not only is it important for orientation in the environment, but we know it's important for survival, it's important for reproductive choice. For example, many animals mate and call, and so sound is important. But more than that, the sense of sound, I've become very intrigued by the role that sound plays in music, in dance, in spirituality, and of course, in sports, not just because of music being in the background, but also because the ear has the task of balance. So here we have Pelé. If you know soccer, you'll recognize the great Pelé doing his signature incredible back kick. The target is here. And he was known for doing this aerial kick into the net, which if you need to go to the soccer archives, I'm a soccer fan, and look at Pelé scoring. He's an amazing athlete. So the ear contains the organs for hearing and balance. Most people don't know much about it. They don't think about it. There are odes to the eye, but no one writes a poem to the ear. And they probably should, even though we have a tradition of song and wooing and courtship. And so sound, I'm not going to talk about sound, particularly sound enters through the external canal, and sequestered in the hardest bone in the body is a complex structure, the ear. In humans, we have both the senses of sound are perceived by the cochlea, and the senses of balance are distributed through other organs. So there's little end organs within the large organ of the ear. And no one's going to give us their ears for research, so in point of fact, much of what we know about the ear comes from studies with animal models, particularly fish, amphibians, chick, gerbils, chinchilla, and mouse. And so this slide really contains core information that summarizes what research about the ear has told us in the last, say, 5 to 10, 20 years. So first of all, and then you're going to see pictures, the ear of vertebrates is composed of multiple vestibular and auditory organs. Even though we think of it as an ear, there's actually multiple little end organs at the end of the 8th cranial nerve, and I'll be showing you pictures of that. What we have learned is that each of these end organs uses the same kind of cell sensor. They're called mechanosensory hair cells. But depending on the organ and the organ's perceptive task, those hair cells become phenotypically differentiated. And that is really at the heart of a lot of the questions that I ask in my laboratory. And thirdly, the biomedical component is where my research is moving into. The ability to repair and regenerate these mechanosensory cells that are the sense cells for hearing and balance is lost as you move up the ladder from chick to mouse to humans, but the regenerative capability is retained in fish and amphibians, which is why we use those as animal models for regenerative processes. So I'm now showing you a picture of a glorious sensory bundle on a hair cell on the left. This is a scanning and picture taken by a graduate student in my laboratory, Vince Lopez Anaya. And what the picture shows you then is the exquisite force sensor that is at the apical end of all hair cells. And you're going to see a lot of pictures in this talk. So the hair cell is a common mechanosensory cell. It is misnamed because there are no hairs here. These are actually a kinocilium with a bulb at the top, for those of you in cell biology. And these protrusions are actually stereovilli. They're not stereocilia. So I work on a cell that is misnamed at many levels, unfortunately. And so how these cells work is that pressure, sound, moves this bundle, and it initiates a signal transduction cascade, okay? And it's damage to these cells that cause loss of vestibular and auditory function, and most of the time. This exquisite, fabulous, beautiful bundle. I love these cells, and when I show you the pictures, hopefully you'll love them too. Anyway, people often think of these cells as, well, we have hearing loss, and I go, no, no, no. These are actually very tough and faithful servants, because although we lose 40% by age 65, in point of fact, our sense of sound, for example, is predicated on the presence of 20,000 cells in each ear, compared to the sense of vision, which is 100 million photoreceptors per eye. So if you start thinking like an engineer, what engineering system is going to last for 100 years and let you perceive sound the way it does? It's a marvel of engineering. They are mechanosensory transducers. They're electrically active. They have ion channels. For those of you that are interested in signaling processes, they have what are called ribbon synapses. A lot of interest in these in the field of neuroscience. And like I said, they rarely regenerate in humans, but they can regenerate in fish and amphibians. So what is the biomedical relevance? Even I was not aware of some of the facts on this. I did not know that sensorineural hearing loss is the most common sensory disorder in humans. Presently, there is a global epidemic around the world in hearing loss due to noise, ambient pollution, drugs, iPods. Sound just bombards us. I'm actually amazed how quiet this room is. It's actually a very quiet room. And so many vestibular disorders, I've been to testimonials where people have vestibular disorder. If you lose your vestibular sense, if you've ever been nauseous, it means 24/7 you're disoriented and nauseous. It's probably more debilitating. And should you ever be asked by a fairy godmother, I'll give you the world, but you must give up a sense, don't lose the vestibular sense. You know, really, that's not the one you want to lose. Probably vision would be, people who have lost them, would be the one that you would choose, although it's not intuitively obvious. It's very essential what we're learning to identify impairments in children because of impact on learning. If you can't hear the teacher, you're not going to be able to respond. And so we're finding is that probably a lot of children are tested for vision, but they're not tested for hearing. That perhaps a child who's unresponsive didn't hear you, the simplest response. And so these are the 5 main reasons of hearing, causes of hearing loss: heredity, infections, acoustic trauma, prescription drugs. Hold on to that thought, I'll come back to that. And presbycusis, a fancy word for getting old, okay? And again, there's a lot of genetics that's emerging about hearing loss, and so we know that genetic factors are important and genes are being identified. So you'll see as I move through my talk how when I set up my laboratory and started working on the system, working on the formation of the ear, which is the question that gripped me, and how these cells differentiate, the genetics has become very important. And I'll conclude the talk talking about some microarray approaches that we're using to really start to uncover some of the genetic factors that may be important in inner ear organogenesis and auditory function. So I'm going to just mention this just to remind you that this is just for introducing the nano word at this point, that mechanotransduction is a nanoscale event. And it's a busy slide, but I want you just to look at the picture on the right. This picture here, a lot of people are really interested in the mechanotransduction apparatus, and I know I was talking to Jing Hay. Where are you? Jing Wei, in the room somewhere. I thought, yeah, you're there, okay. You're behind there. Just that, you know, we were talking about mechanotransduction being important in plants. And what we see with these processes that are transduction, the membrane is involved, and then there's things happening outside and things happening inside, and there's pressure transduction, and forces are sensed. And we know that many of these events in the membrane can actually eventually end up directing and affecting gene expression. So what we know then, or what a lot of thinking is done, is that as you move the bundle, what happens is this channel is mechanically open. It's linked to another bundle, and it's actually physically gated open. And then due to an actin-myosin interaction, there's a slippage event that then closes, which is why you turn the sound up on your iPod, and after a while, it doesn't sound as loud. It's adaptation. It's what the nervous system does. Okay, but the hair cells are still perceiving that intense, and you can shear the bundles. And so this is the final slide of the introduction. This is a crash course in hair cells and the hearing, is that the hair cell, I see it as a neuron. It's a neuron in so many ways, and as we start to think about it, It is filled with ion channels. It has neurotransmitters. That's the criteria of a neuron. You make neurotransmitters and you release it on postsynaptic cells. And it's a very truncated, morphologically abbreviated neuron, but nevertheless, in terms of the function and the components, it is a neuron, and I see it that way. And because I'm a biophysicist and an ion channel person, I'm very interested in these channels, and we focused a lot on trying to understand these ion channels in the lab from a genetic perspective, and I'll talk about that in the next slide. So, in this part of the talk, I just want to introduce my lab and go through some cool pictures of the ear and kind of give you a sense of what we've been doing. I'm at New Mexico State University. It's a Hispanic-serving minority land-grant institution. It has an engineering and agriculture mandate for the state. Here is the Oregon Mountains that are near us. These are pictures of my lab partying at White Sands. And then this is our building that's being renovated, and this is a person in my lab, Miranda Salt, who is working in the lab part-time as a technician and pursuing her undergraduate degree. So we're a neuroscience laboratory. We're often asked to identify ourselves. I really hate doing that because I work on process and I hate nouns that are labels. But these are some of the words I put down. Sensory systems, development and regeneration, nanobioscience and biophysics, these go together. In fact, nanobioscience is really a fancy word for biophysics in my opinion. Bioimaging has become a theme. It was not a theme I had. That was an expertise I developed while I was there. And of course, the genetics of the nervous system. And these are examples just illustrating the kinds of approaches that we use that are molecular and also imaging in the research. So it's a pretty slide. It's on the webpage. I want to introduce the people whose work I'm going to be talking about. I hate leaving people to the end. They kind of get rushed through, so I'll introduce them now. This is us at, I think we fed. Yes, I think we fed. They look like they fed. Okay. And we feed and sometimes we eat, but in my lab we feed a lot of the time. I think we were really hungry, so we're going to be feeding. And so this is my laboratory, and from left to right, these are the people whose work I'll be talking about. Tushan Powers is a postdoc. She's doing the microarray work. David Seltzemeyer is doing the promoter work, and he's single-handedly set up a lot of cell culture systems. Lina Urquidi is working on our collaboration on optical coherence tomography. Casilda Provencio runs my lab. We're joined at the hip. She does a lot of the molecular work. She's the molecular queen. Marty Morales is a PhD student who's doing an innovation project, and Eddie Lamas is a new RISE student in the lab who's working on the organ cultures of the ear. We'll be talking about their work. So these are the core questions that motivate my lab. First of all, and this is a big sort of development question, how does the tridimensional, because it really is a structure that must be understood in 3 dimensions, inner ear arise from the otic vesicle? And I'll be showing pictures about that. And specifically, I think the second question is the one that really drew me to the field as I started learning about the ear, is how do hair cells acquire structural and functional phenotype within sensory organs during development? And the structural phenotype that I'm talking about is the diversity of hair cell bundles that's specific to the end organ. And the functional phenotype are the ion channels that seem to be upregulated as cohorts in particular organs, and is coupled to the fact that different organs are specialized to pick up certain frequency modalities. So we have low frequency organs, high frequency organs. So when you start talking about the ear, you get into the world of physics and frequency and that very quickly, which is why most people who work on the ear are actually biophysicists, engineers. It's very rich in that field because you need to get into an engineering mode. And then finally, where my research is moving to, what I'm very excited about now because we've laid the basic science foundation, is really starting to develop projects that now take this basic information about a system and start looking to ask the question, can we identify mechanisms? Can we identify interventions that promote the restoration and repair of hair cells? Okay. So that's the area that we're going to. And so you'll hear me coin this word, electrophenotypic differentiation of hair cells. And what that means is different hair cells are going to have specifically different ion channel cohorts in their membranes because they have to respond to a best frequency. And so that's a big question. I don't know if we're going to get it answered, but it's kind of like, why not have it? It's easy to have a big question. It's a small question, so let's have a big question. And then within that, we ask smaller questions. So that's the big question. Yes, please.
What is the nature of the
the nature of the endolymph, and you're so astute, because that's actually a very sophisticated question and tells you, okay, so this is a system that's hugely complicated. The endolymph bathes the apical end, and it is high in potassium. And the perilymph is basically what we have in most of our fluid bodies that you think of, and it's high in sodium. And so when this tissue forms, as it turns out, all these, you're not getting sodium entry like you would normally. So the hair cell, this is a deep question. We can talk about that later because it really is a deep question. So when you study the hair cell, you have to understand that this is a cell that lives in two very different environments. High potassium on one end and high sodium on the other. And what that means is that most people who've done physiology have not done the physiology that accurately represents what the system is like in vivo, because it's hard to generate physiological models and probes that let you do both.
I think of lymph, though. I think of lymphatics.
Yes.
Is this anything related?
Yes, it is. And in fact, there's a lot of interest in the immunology of the endolymph and the immunology of the ear. And yes, there are globins. You see it in some of our SEM pictures. People who have studied the ear, who have studied other systems, say it looks like there's globins and things that might be discrete. But not that many people work on the ear, and after you see the talk, you'll see why, and you probably won't either, because it's a really, really hard system. So that's a great, great question. Okay, so our research approach, I look at it as two levels. Mechanotransduction processes are at the nanoscale, so we have projects that are related to signaling networks as processes that underlie transfision and reception, and that's where we're interested in developing bioinformatics types of projects. And on this scale, we're talking about ion channels, cytoskeletal proteins, quantum dots. I'll be talking about that at the end of the talk. And then the larger question is the question of how the organs form during development. And that is a big question, and so my interest in doing systems biology last year is to really think about that as a systems process. And it's a different way of thinking for those of us that work as cell biologists. We're used to gene by gene, cell by cell, but really to try to develop modeling and ways of looking at large-scale processes. And it's an emerging field. It's not systems like, we've always done systems. Everybody works on a system. It's something a little more. It's about the integration of mathematics, physiology, and molecular biology to establish predictive models that you can then test in the laboratory. So it's a little more refined. And so this is like the new stuff that we're doing, the tissue culture, the microarrays, and the bioimaging. That's the latter part of the talk. We work on Xenopus, a familiar model to this group. I don't think I have to say too much about it. It's an NAH model organism. We work both on Laevis and on Tropicalis. And the reason I worked on this model is because when I went to New Mexico State, I knew I wanted to identify a question that was unique, a field where nobody else was working on, because you need to establish your own identity. I had worked on hair cells at UCLA, and I had worked on mouse models, but there were no facilities there that would support mouse models. So I said, okay, development, what would be a good other model? And across the lab where I worked, people worked on Xenopus, and the more I thought about it, the more it seemed a logical model for developmental biology, because much of what we know about hair cells was done in amphibians. And so basically my lab pioneered the use of Xenopus for these studies, and I think the payoff has really come together. And the reason is that, like I said, amphibians are a standard hair cell model with regenerative potential. If you look at a picture in a textbook of a hair cell, it's usually a hair cell from a frog. It's the Hudspeth, it's the person that founded the hair cell biophysics field. Much of that work is frog. It isn't a mammalian model. And there is actually neuroethological research on Xenopus, and we know something about how it hears as well. But the main reason for working on Xenopus, other than the development, is that the fundamental, much of what we know about signaling in the ear has been done with amphibian models. And of course, the payoff was when the genome was sequenced. And so you can now, if you work on Xenopus, go online, and we're having so much fun with the genomics that's out there now, because the Xenopus genome has been sequenced, and this is a huge, huge boon. And until your organism's genome is sequenced, you don't realize how valuable it is and what that lets you do. Because you can do thought experiments with the genome and matching. It's really, really a lot of fun. So this now, I'm going to start showing you pictures of the ear so that you see why this is so hard to work with and why it's so intriguing. So this compares a labus and a tropicalis ear. This is a piece of paper, that's 1 millimeter. So the first thing you see is that this is really a tiny structure. These are the ears of post-metamorphic animals. We're having a ventral view of the top of the head. And so you have the 8th cranial nerve, and this structure here is the ear, okay? And the ear, your ears, our ears, have calcium carbonate crystals, and they exert load on some of the vestibular structures. So our ears are packed with calcium. And so if you do molecular biology, you know that that is not necessarily something that you want. When we started working on Xenopus, little was known about the organism, so the first thing we had to do, since we were interested in development, is figuring out how does the ear grow? How does it develop? What do the structures look like? So we spent a lot of time doing that. And what the anatomy illustrates is something that is important for systems research, the reason why systems approaches are interesting. And that is the biocomplexity, that this is a hugely complicated structure. And that if you want to understand genetic and regeneration, you have to do it in the context of what the ear is doing as it naturally develops. And so we did a lot of anatomical research, and in parallel with that, we started long ago, like in the 90s, initiating molecular investigations of genetic determinants that underlie organogenesis. And this was really, really, really hard. I think in my naivete, I had no idea just how hard this would be. I mean, this, I think, was the thing in my lab that took us so much time to do. And it wasn't just our lab. Many laboratories that work on the ear face the same problem. And so a lot of what we did, and I would say this is probably a long term, I would say almost a decade, was developing methods for molecular investigation. And that, of course, was in parallel with new tools and resources that the companies were putting out. So the equipment and the materials you have available are far superior now. And so let me tell you why it's a challenge to work on the system. First of all, that's an ear over on the left, that's a millimeter. You dissect this thing out, and it takes people a while to learn how to identify the structures. It's a complex structure with multiple sensory surfaces, high levels of calcium, surrounded by the hardest bone in the body. In Xenopus, we've had to use a dentist drill with some of the adult animals to pull the ear out, because the bone in the ear is 8 times harder than the rest of the bone in the body. The tissue is incredibly difficult to work with, to get good nucleic acid and protein extraction methods to work because of all the calcium, and because the tissue itself has just a lot of components in it that I think reflect what it does. It makes it hard to do DNA and RNA protein work. Tiny amounts of tissue. 1 ear might give you 1 to 5 milligrams, so you have to sacrifice 5 to 10 ears just to get 1 to 2 micrograms of total RNA. So that was a challenge. There's very low numbers of the target cells. There's only 15,000 per ear. And then the question that we're interested in, there's small populations of cells next to each other that are the target cells. So the target cells are few in number. So what I'm going to talk about in the next couple of slides is just to show you examples of some of our results. And this is work that's out there and work that's getting out there now. We have a lot of papers coming out right now that are in press and other ones being submitted. This was a long-scale project, and students were essential for this work because it was a long-scale comparative work. So what we've done, and I'll show you some pictures, is we've shown the structural differences between vestibular and auditory organs. We have established that morphogenesis continues after metamorphosis in Xenopus. Things continue to grow and add on, and that has implications for neurobiologists. The patterns, we've shown the patterns on the sensory epithelia as the sensory cells mature, and I'll show you pictures in a minute. And each end organ, and there's 8, follow a different timeline. They're all developing in slightly different ways. And some of them stop developing at metamorphosis and others don't. So even though the ear is one organ, within the ear, different kinds of things are going on. And even though there's hair cells in all of them, they're subtly modified in what they do and how they develop based on where they are. And then a lot of what we've done, and this is a complicated, a lot of detail, that's a whole seminar, I'll allude to this. We've started doing a lot of promoter work. We're really interested in regulatory transcription factors that could activate groups of genes during organogenesis. And so we've actually pulled sequences out for promoters and transcripts. And Our future directions, because this is now the rest of the talk, what we're going to, having done this has given the foundation for what we're going to be doing in the future, which is working on cell and organ culture systems. And now, a lot of the research in my lab will be collaborative, and I really think people are hearing this, we need to collaborate, it has to be interdisciplinary. I think it's essential for the questions that most of us want to answer. And so I think this is actually an exciting part of the work, but I think I cannot go to the next level in my research without doing it collaboratively. And then finally, I've had, Tushan is my first postdoc. It's been undergraduate and graduate students that have been participants in this research, and they've done a fabulous job. So let me quickly talk about inner ear structural development. I think there's interest in bioimaging, so I'm just going to show you some pretty pictures. This is an array of the different kinds of imaging methods. If you do microscopy histology, you know that everything you do, well, it's science, right? Everything you do gives you a piece of information but introduces artifact. Histology can introduce shrinkage artifact. Different kinds of imaging gives you different kinds of information about the structure. So we attack the ear from many different directions. So we've done TEM in the lab, scanning EM, we're doing a lot of confocal microscopy, doing tridimensional image rendering, and then of course your standard section tissue, and of course, again, more confocal microscopy. So we basically do the whole array from section tissue to whole organ, TEM, SEM, and confocal. So let's get more detail into the ear. I've told you it has multiple organs. This shows you the nerve, the eighth nerve coming in. When the eighth nerve comes in, that's the cranial nerve that innervates the ear, it branches. And in the anterior end, you have, this is what's highlighted here are the auditory organs in purple. In the anterior end, you have two canals. These are the sensory fields, and the utricle. The utricle is the gravity sensing organ of the body. The utricle has crystals on it in our ears and in Xenopus. And the sacculus is that big one here that has crystals on it also. And you'll see another picture in a minute, a zoom picture. Posterior, you have your auditory organs and another canal. Because remember, the ear has labyrinths, and I've become more fascinated with those. So the labyrinths of the ear are the Cartesian coordinate system in our head. It's a beautiful thing that what the ear does is it gives us that sense of XYZ position. And that's the task of the canals. And again, this material often is in touch when my students go to med schools, they end up teaching the ear in their anatomy classes because even the professor will go, well, I don't know the ear. It's complicated. So in Xenopus, we have two auditory organs, the basilar papilla and the amphibian papilla, and 5 vestibular organs, the canals, the utricle, the legana, and 1 organ, the sacculus, this large one, that serves acoustical and vestibular function. This is a summary slide that's years of work. And so we've looked at the sensory fields of all of these. So these are scanning pictures. And what the picture should show you visually is part of the marvel of the ear, why understanding this organ is so complicated. These are the sensory fields of the organ. So the first thing you see is 1, the pattern of the field is different. 2, the numbers of cells is different. And 3, the bundle shape is different. So when we build an ear, you're to build an ear, you have to come up with a model that patterns each organ with a different sensory field, and within the organ directs the bundles to become morphologically specified. It's pretty cool. I can, if you're an imaging person, you're going, oh my God, so exciting. And then here is a picture just showing how great confocal is. That's an SEM picture. If you do SEM, it's like forever to get these. You might do 20, you might get 1 reasonably good picture. Confocal, that's a 3-hour prep. So we couldn't let the SEM people see the confocal because it was just too depressing for them to see what you could do. And I'm just for fun, and because I've been talking with you, I'm going to run a movie. So this is a confocal projection. This is the basilar papilla. It's the highest frequency organ. These are the hair cell bundles, these triangular shapes, and this is the confocal projection. This organ's like a donut, and the hair cells sit here. The nerve would be coming in, here are the hair cells. So this just shows you how complicated this tissue is. If you're scanning down the stack, do you see how there's a curved surface? So not only is it a pattern in terms of shape, it's a pattern in terms of it's a curved surface. And so I'm very excited. I actually have hooked up with somebody who's a mathematical modeler, and we're going to try to do nonlinear differential and equation types of analysis to see if we can pattern different kinds of sensory
We're
fields.
supposed to be seeing something?
Pardon me?
You're saying there's nothing.
Oh, well, no wonder you guys can't see it. Okay, well, let's see what happened. It died. Is it back? Okay. Maybe it was just a connection. Let me see. Did you see this? Did you get this much?
Yes.
Was it when I ran the movie? Okay, it doesn't
Yeah.
like the movie. Okay. That's fine. No movie for you. Hmm?
It's pretty on your screen.
Yeah, it's pretty. Yeah, those people behind me going, yeah, it's really cool. Okay, anyway, sorry you can't see the movie. Okay, so this is now going to take you through a tour into the ear. This is a Xenopus larva. So at this stage, the ear is mature. And we're going to take, it's like if you saw the movie Powers of 10, it's my contribution to that philosophy. So here's a Xenopus tadpole. The ear is in this region here. We're going to start zooming in. And as you go in, what you see then are the otoconia. So here's the utricle and the sacculus developing in an orthogonal relationship. Okay? And if we zoom in some more and we dissect the ear out, this is the blob you come out with. And if you're in my lab, you'll learn how to distinguish detail. But this is the nerve. We removed the calcium in this opaque area. The sensory cells are at the end of the nerve there. This then is that cartoon I showed you. The nerve is coming in, and these are the different structures. We removed the sacculus. And so the organization, I've shown you scanning, but in fact, the ear is made of epithelial tissue. Okay? And so if you section any of these organs, what I've shown you are the scanning pictures at the top. You can see the little bundles here. But the hair cells themselves are in the epithelia. And part of our field's interest is what gives rise to new cells? Is it through mitotic or non-mitotic regeneration? Or is it transdifferentiation of a supporting cell? And I'll show you one slide about some of our work in that area. And so you would see this, and again, that's 25 microns there. And then when you scan, depending on the organ, you would then see on the surface these mechanosensory cells, and they're embedded in a matrix. So there's like a mosaic. They're surrounded by supporting cells, and within it there's a hair cell. And for people interested in cell biology, there's a lot of interest in the asymmetry at the apex of the cell, and how you have the tubulin establishing polarity at the surface. And
Are those kinocilia the long?
yes, yes, they are. And so I don't have time to talk about the difference between humans and Xenopus. I mean, this is like a huge, for most people, they're so unfamiliar with this system that this is like, there's a lot you need to communicate to show that. But in, like for example, in the cochlea, we don't see that. If you've seen the pictures of the cochlea, there are those 3 arrays like that you see in the books. The human cochlea is organized differently. So it's thought the kinocilium emerges and then dies back. Okay, so that's these are the stages we work with. We had to go through and kind of, okay, how are we going to stage? You know, when you do staging, you know, animals are like people. Some of us are tall, some of us are short. Are we all 18? Are we all 45? What does that mean? So we had to address that. So we picked stages where we could really distinguish the different animals. And what I want to give you the feeling for now, I've shown you that complex structure. But the incredible thing about studying development for those of us that work in this area is this fascination that the ear started as a plateau, becomes a vesicle, and out of that vesicle, you get that incredible structure that I showed you. And so we've done a lot of work showing this is how you start, sensory fields start emerging. These are the canals starting to form. What happens, how do you form a canal? It turns out that what happens is the walls invaginate and they fuse. And that's how you form the labyrinths of the ear. And so Quincy Quick is a student that did a lot of this work. And for those of you interested, if you want, we can talk about that. We've become very interested in doing a lot of work on where cells are emerging using BRDU, looking at mitotic regeneration. So what you see if you label with phalloidin is you have a central field where the cells become differentiated, but mitosis is happening on the periphery. So you're growing from the edge out, doing some. Okay, so basically that otic plateau gives rise to all these compartments. My lab has done this work for the ear, for Xenopus. Mainly between 37 and stage 50 is when the ear forms. So that's a couple of weeks. It happens somewhat quickly. The hair cells differentiate from precursor cells in that epithelia. And in Xenopus, the ear continues to grow and hair cells are added throughout animal life. And the work I'll show you in a few minutes, we've also shown that the hair cells can regenerate. Marty is a grad student in the lab, and so where we're moving to now, because of her interest, is we started looking at synaptogenesis. So Marty started looking with different kinds of labels at the nerves coming into the hair cell, the kinocilium's labeled, And the cool result that she got from some TEM is she found evidence for ribbon synapses in Xenopus during development. So she's got a really cool TEM project going on. And that's all I'm going to say about that. So our anatomical work right now is pretty much wrapping up. We're really starting to do other kinds of things. The synaptogenesis is something where we have a little bit of interest. So now I'm going to switch gears and talk about genetics and talk about ion channel promoters and expression. And so mechanoreception in vertebrates. When you initiate a signal transduction and you're part of a neuron, that means you enter the world of electrochemistry. You've got to initiate an action potential or some kind of event, and then you've got to get neurotransmitting release along the signaling cascade. And so in vertebrates, what we know is that frequency encoding is dependent on the ion channels present in that membrane of that hair cell. And just like I showed you, heterogeneity in the bundles, we also know that there's heterogeneity in the ion channel complements. And so this is a slide from a colleague of mine, Peter Nairns, he's actually a mentor, and a student, Mike Smotherman, where they went into the amphibian papilla, that's one of the structures I've shown you, and showed that depending, and others have shown this too in chick, depending on where you are along the sensory field, you have more or less of different kinds of ion channels. So now, you want to build an ear, not only do you have to understand how the bundles form, but you also have to understand how is gene expression regulated so that particular ion channel genes are turned on and turned on in certain numbers. And so this is going to be true for every organ. It's a really big question. So I became interested in this question early on, and as an ion channel biophysicist, I said, you know, we all keep patching channels. How's that going to let us, how is patching channels, which I love, by the way, going to teach us anything about how channels are expressed in cells? And so for those of you who are students, you've probably seen this kind of slide before, and it just shows basically the central dogma. And if you're interested in ion channel regulation, you can work in different places along the cascade. If you're a patch clamper, you're probably working here at the protein level. What factors affect ion channels and the turning on. But in neurobiology, unlike plant biology, plant biologists have this edge, we know relatively little about promoters. The promoter interest in the nervous system is something that's emerging now. So I became interested in ion channel promoters specifically. And so we started cloning promoters in Xenopus and cloning the transcript for a particular channel. And so we've worked at different stages of the cascade of gene expression. We are interested in identifying inner ear transcriptional control elements. So we've cloned promoters with Genome Walker, and we're making fusion proteins for heterologous expression. That prompted the cell culture research we're doing. And then we started working with transcripts as well, trying to clone full-length sequences as well. So this has taken forever. This was so hard for us to do. And recent advances in RNA isolation methodologies and also just the kinds of enzymes that are available are such that this made this possible. Our genes are big. They're 3, 4, 5 kb. And people often clone pieces and put them together, but we went for the goal, end-to-end cloning in one. And that was hard, but it's done. And then expression cloning of the clone channels, and we've been doing that in collaboration. So we've cloned our ion channels and expressed them in collaboration with somebody at Utah State. So this is our favorite channel. I'll just mention it for those of you who are channelologists. We're interested in the calcium-activated potassium channel, also known as BK, also known as slowpoke, also known as KCA 1.1. This channel defies everything you've ever been told about genome organization. First of all, on the coding end, it's alternatively spliced heavily. And on the promoter end, it doesn't have a TATA box or any of those things that we're taught. In fact, it's like a locust that is cut and sorted. So this is a picture from a Drosophila project that the Drosophila people have actually cloned this and figured this out. So that this is a huge upstream regulatory region. And you find control regions that then direct the tissue-specific expression. We happened to pick not just the hardest system to work on, but the toughest channel. We knew it was important from the physiology, but nobody had a clue that the upstream organization would be so complicated. So we're working with this channel. And I'm just going to tell you the results of the genetics. We can talk about this. This is a lot of work. We've cloned the promoters both from Laevis and Tropicalis, and we're making fusion proteins to try to see if we can direct the expression, to see if we have the regulatory element. And what we're really interested in the bioinformatics approach is, this sense, can we start doing large-scale promoter analysis to see if we know there's a lot of genes that are turned on, can we then start aligning promoter sequence and trying to sort of reverse determine regulatory elements that might work in patterning tissues where things work together. This is an example of what David has done. These are 3 channels that we know work together. So he's been doing having fun with the bioinformatics, looking for these transcription factor binding sites. We know that these are promoters that are expressed together in the ear. So what are examples? Can we hypothesize from the bat? Are there certain transcription factors that might then all work at the same time to put these together? This is really the bioinformatics that I'm really going to plug. But we can start now thinking and hypothesizing based on looking at all that fabulous information that's there. We've also cloned the gene transcripts. This is the work of Priscilla Provence in collaboration with Peter Rubin. We've actually shown expression of our channels, so we know we have full-length sequences. And if you're interested in more of the, this becomes very technical quickly, we can talk about that later. Because what I want to spend the last part of the talk is talking about our current projects and future directions, and talking about cell culture systems, regeneration, systems biology, and imaging. So, about 4 years ago, I knew that we were quickly going to need a way to test and manipulate our system. And so I've had a lot of experience with cell culture. So we started developing Xenopus culture systems. And we did a cell line to get the culture system going. Then we've set up dissociated neuromuscular cultures for looking at some of the genes that are in neuron. And then the hardest for us, and what I want to focus on in the next few slides, is developing an organ culture of the ear. We're hugely excited because we've got that to work. So we worked with the Xenopus kidney cell lines. It turns out there are Xenopus cell lines, the A6 cells, and so we established cell culture methods in the lab. That's what we use to train students and people in aseptic method. And we use these to develop transient transfection methods with lipofectomy. So we optimized with the cell culture. And then we went to the next level of complexity. People have cultured Xenopus neuromuscle for a long time. So we set up Xenopus neuromuscle cultures. And these are intermediate systems. And we've been able to get transient transfection of these. So everything's working nicely. We've got the appropriate expression of promoters that we know, that we clone, that we know are expressed in neurons. And the third system that we set up, and this would be like the work of the last 3 years, are Xenopus organotypic inner ear cultures. Because with an organotypic inner ear culture, we can then go in and actually manipulate and do some pharmacological interventions. So David Saltemeyer set up the A6 cell lines. I'm just gonna mention him. I'm not gonna talk in detail about these. It's great. It's a good way to set up the transfection methods. And this just shows you, just great pictures from the cultures. These are the Xenopus cell lines. And he made the PEGFP actin construct, and then you can put the Xenopus promoters in there and drive expression. So he showed that you could get the transfection to work, and then you could exert actin promoters from Xenopus laevis and tropicalis and drive expression. So the proof of concept of taking a promoter, infusing it, and doing a transient transfection was established. He also then, this is a grad student who feels he doesn't work very hard. I keep saying, you've got to finish and go. You've worked enough. He's wonderful. And he's finishing up this year. He then set up the neuromuscle cultures. And I'll just show you this one slide. These are examples of the cells in culture. He's immunologically characterized them. So he has muscle, he has neuron, and we also have epithelial cells. And he did a really nice project. I'm not going to go into the details. He cloned a couple of genes that are expressed in muscle and neurons and did a transient transfection. If you're interested in this, I can talk about that later. So now I want to really spend time talking about what we're doing now, what we're excited about. So we were really interested in developing a system to probe and perturb and to see if we can have a model for hair cell regeneration. And in particular, we were curious in starting to look at whether hair cells can regenerate after ototoxic insult. So Shannon Manolita is a master's student in the lab who took this project on. And Eddie Llamas is an undergraduate who's now validated her results in the last couple of months. And so what Shannon had to do is use what we learned from the other culture systems and take the whole ear out and get it to survive in culture and characterize it and validate the model. You can't use antibiotics, so all of this has to be aseptic because it turns out most antibiotics that we take damage the ear. Gentamicin, streptomycin, cisplatin, I'll show you a slide in a minute. It's actually amazing when I started to look at how many drugs all of us take potentially damage the ear. I don't think that knowledge is appreciated as much. So what she did is developed the culture system and then characterized the viability. And we did two things. We would put the ear and just let it float. It became a ball. And then we said, well, it's a mechano organ. What happens if it maybe needs to be anchored? So then we used cell tack to anchor it. And Shannon actually got these cultures to work 14 days beautifully. So what do they look like? Well, how do you know you have a successful organ model? Well, you have to validate your model. So do you have membrane integrity? Is there metabolic activity? And then, of course, for us, is hair cell bundle morphology retained? So we have a good indicator of whether the culture system is viable. And so we did a whole array of characterization using a lot of fluorescence microscopy. And the end result is, after Shannon tweaked the system, is that both the floating and anchored cultures are viable, but the anchored are superior. And so this just shows you, this is 5 days of what it looks like in the floating culture. What the floating culture is, the white would be actin, so you see the ligane and the hair cell bundles. The cultures stay alive, but the tissue deteriorates, okay? You get tissue shrinkage and curling. But when she uses cell tack, you get this beautiful result. So here's a 10-day culture. Here's that scapular sensory field looking beautiful. Here's the nerve, red, still viable, because in Xenopus, the sensory ganglion is right next to the sensory field, so you actually have the nerve there together. And then when you zoom in, you see a sensory field that's indistinguishable from a 10-day animal at that age. So then, remember I said prescription drugs, gentamicin, streptomycin, tobramycin, chemotherapeutic drugs. All of these are things that we take that actually can destroy hair cells. We then challenge the cultures with an ototoxic drug. And this is a list of examples of drugs that we now think could affect and destroy hearing and vestibular. And so cisplatin, for example, can cause vestibular and auditory damage. And so many, many, many chemotherapeutic drugs have a very adverse effect on the ear. So what we did is when you get those cultures, gentamicin, after 48 hours, the bundles are gone. It completely destroys the bundle. And then, when she lets them grow, this is what it looks like after 8 days. So we're hugely excited about this particular result because we have a model for regeneration. And Eddie Damas in the lab just replicated her result. I was very excited. I said, no, it's one person who knows. I need to see this done again. And he's replicated the results. So we're very excited about this particular. It could be potentially a really quick and easy high throughput model for looking at drug testing. It's Xenopus. You pop the ears out of the larva, boom, boom, boom. So, and then you move to another system. But I think it's going to be a really useful screening model. So anyway, in terms of the results, the lipofectomy transfections work. Transient transfections aren't super, but they do work. We're able to get our promoters to drive expression in culture systems. And then we have a great culture system that we can now go in and look at, hit it with BRDU, is there mitosis, and really characterize that. So I want to mention something about systems biology now. Last year I went to MIT, and I'm very excited about this. It's a systems biology roadmap center. The head of that is Peter Sorger. And the thinking behind this is that we need to come up with new ways of approaching the problems many of us have faced. We've all been doing this gene by gene approach, but the complexity is going to escalate as we start looking at different kinds of questions, so that we need to come up with different ways, and Peter put it really well. Bigger and faster computers aren't going to make sense. The barrier is going to be crossed by, and I love this because everybody can play here, creativity. Anybody with a good idea. You don't have to be in the university where you have $1 million core lab. You just have to be able to think and put things together and model and take this data. And I'm very excited by that because, as we all know, the funding's going down. But hopefully our brains are still intact, so all of us can ask good questions and model. So it's really really starting to ask questions and work in collaboration with mathematicians. And so what was interesting also to me, and with regard to organogenesis, and I expect, and I've talked to people here, many of us actually work with tissues that are making a decision as a gen work. Do I live or do I die? And so you can identify, depending on your system, like whether you have pro-death networks or pro-survival networks. And so what this center is doing is using cell cultures and different kinds of growth factors, looking at how cells make decisions. Because sometimes a cell has to decide, do I live or do I die? And what factors influence whether a cell lives or dies? And that's very important in cancer research, and it's also very important in regeneration. And it turns out that, so they're looking at EGF, IGF, insulin receptors, TRAIL and TNF, which are signaling pathways many of us also think about. So they've got a cool website. I recommend people looking at these. And pertinent to this particular position that I'm interviewing for is that what this is like the research teams of the futures. It's a team model for interdisciplinary research. So this systems grant is the equivalent of 6 R01s. It has a strong outreach component because it's mandated of these large grants. It's like a $20 million grant. It has core component, and it has a huge junior faculty development component as well. They are mandated, as NIH asks us all to do, so they have wiki. They use the wiki concept extensively to collaborate and do activities. And there is a meeting that happens of the participants that's run by the postdocs and students. So there's a lot of ownership of the concept at all levels through the system. And it's organized around really that this is an opportunity for junior faculty to move forward. So I'm very excited by this concept. And so I'm trying to take some of these ways of thinking back to my lab, and the idea is that we would combine molecular biology, physiology, and computational modeling and data mining to perhaps identify genetic subnetworks within that are involved in a large scale. And I think to do this, if you enjoy math and abstract reasoning, our temptation is to put all the proteins together by terms of interactions, but that you may have to step back and say, there's processes going on, and that it's the network may not be a network that's so much a network that you can track everybody interaction, but that they work as a group of genes together. So it's it's a different way of thinking about systems. So I'm just going to tell you a little bit about the microarray project because we're hugely excited. So these are 3, I took my students with me. We had a lot of fun. They have a lot of resources there. We learned a lot. I'm very interested in the proteomics. That would be the next level to start integrating. And I had fun talking to the young faculty there doing proteomics. And the main thing that we did there is we arrayed organs. And I said, okay, they're arraying cells. Let's array organs and start looking at the organ transcriptomes. So here's Xenopus. We took brain, ear, kidney, and actually the kidney cell line. And a huge part of our time, if you're doing microarray, and I think that Godman knows this very well, you've got to have high quality RNA. And we spent probably 6 months getting RNA. And there's a tool that you need, and that's the Agilent bioanalyzer. That has been the core to our cloning success, because now it isn't just like a gel, yeah, it looks like this. You get an electropherogram, and you can see the integrity of the RNA. And so we had very stringent criteria. We did not array unless that RIN value was above a 9.2. And we just did it over and over. And then you make the cRNA, you array, we did the AFI chip, it was a core lab. And then we did replicates and triplicate. And now we have masses of microarray data that we're just spending time working with. And part of what we have to do is curate and annotate the chip, because it's a Xenopus chip, so a lot isn't known. So there's projects now that are bioinformatics. You have the probe set, you have to figure out what they are. It's very exciting for the lab. Casilda was the one that isolated the RNA. This is where we would start. It would look like this, totally degraded, but it had to become this for us to be able to array. And we had to go through kits. And what's amazing is if you run this on a gel, you can't always tell it looks like that. But the ability to use this, and this is lab on a chip technology, which is just so cool, all the miniaturization. And so this is the array. I'm not going to go into the details, but look at the nodes and nodes. What you see is the kidney separated from sensorineural tissue. That's the first node. So when you array kidney, you can see on the chip. I mean, it's what you would predict, but it's really cool to see it on the chip. Kidney is real different than sensory neural tissue. And then the next node is, these are ear, and this is brain. So then the chip can tell you, I'm brain or I'm ear. And then the cool result, the one that's really exciting to me for organogenesis, this is larval ear and adult ear. The chip could discriminate development. So, you know, we have other things that we're doing. Let's say the A6, and then if I put the A6 cell line on here, it separates from organs. So that would separate completely different. So cell lines are completely different than organs. So we're having a lot of fun doing the analysis. And here is just one kind of analysis that Tushan did. She pulled deafness genes out of various lists. Again, you have to go to the literature, pull the deafness genes out, query the chip, pulled out the deafness genes on the Xenopus chip, and then she sorted the data. So these are the organs, okay? These are the ear genes in the different organs, and these are the other genes. And if you group the genes that are not ear genes, they all look the same. But if you look at the organs, the ear deafness genes are higher expressed in ear tissue, which to me validates the methodology, because it shows, I mean, it's what you would expect, but to see it on the chip, I've been reluctant to really believe in chip technology, but now I'm going, yeah, the chips are good. Okay, so basically, we're doing a lot of microarray approach, and so we're going to continue. We now have to go back, because once you get the chip, you have to go back with qRT-PCR and validate. Lots of curation of the chip, and the next generation of Xenopus chips is going to have 100,000 probe sets, so that's going to be. So the last part I'm going to talk about now really quickly is collaborative technical projects. So when you hours, they can't work on the project anymore till the next month, but it's a small company and it's really interesting. And so this just shows you some of the examples. OCT is kind of like ultrasound, but it uses electromagnetic waves. If you're interested, I recommend you look at that. Here's a tropicalis ear section. Here's a labial, a little bigger, and this is the OCT image. And this isn't even high, you know, it isn't tweaked. They were doing proof of concept. So this was an animal that was imaged, and you can start to see the ear. And so depending on how, and then the light that they use is infrared, which we all know is a really good wavelength. It's 1300 nanometers. So we're working with them. They're doing instrument development. You go up there. It's all a bunch of physicists and spectral people, and it's really cool. And they need student interns. So there's actually opportunities for students to intern in industry labs, which I did not know existed. And this is the really cool thing, also excited. It can take right now, because of their system, it might take like about an hour to scan the whole animal front to back. But because it's a digitized image, this is a transverse plane, so if you're a Xenopus, you're scanning like this, but you can digitally reconstruct the sagittal and coronal plane. And that is totally awesome. And so you can get into the world of animation and three-dimensional rendering. And so they're looking for resources. And of course, the eye is just always so spectacular when you do anything. It really comes out nicely. And here's the ear. Remember I showed you the canals fusing? You can really see that. So here we're sectioning with a knife. And here, there's this. So moving to live animal imaging is something we're interested in. And now, the last thing I want to mention is work that we're doing now in collaboration with the Center for Integrated Nanotechnology. So, quantum dots. These are colloidal semiconductor nanocrystals. And they are fantastic as probes because they have very high extinction coefficients. They absorb a lot of light. And they are very photostable because they're a metal, okay? And so I became interested in these as probes that give you more signal and also let you image with lower light intensities. And we actually did a project looking at the proof of concept of combining them with multiphoton imaging. So this is a picture. There are 10 to 30 nanometers. You make the coat. You have the core that's heavy metal. And then they have a coating because you've got to start to solubilize these things and stabilize the core. And then this is where you work with chemists. This is really interdisciplinary. So you'll go in, you have a chemist who knows how to make these particles, and then they go, well, what do you want on the coat to make it compatible with your tissue? And you can come up with some concepts. And then the organic chemist has to come in and work to put this together into link bonds. It's a really fabulous interdisciplinary project that you cannot do this without having your biologist knowing receptor ligand, your chemist coming in and doing the chemistry, and you know, it's just wonderful. And so it's really a lot of fun. And these are, they can fluoresce when you excite them by light. And because they're all made of the same material, all of them have the same spectrum. So what happens is that the size determines the emission properties. And so the bigger it is, the more it fluoresces in the far red. And the smaller it is, the more in the blue. And so these are the spectra of what they look like. They're all tends to be excitable in this range from UV to blue. And then the bigger dots fluoresce more and more in the red. So you can do multiplexing. You can use the same light excitation and then have lots of dots of different colors and look at lots of different target molecules. So it has a lot of potential. And it has advantages and disadvantages. We actually compare them to Alexa. They're fluorescent and electron dense, so then you can do TEM after you use them. So that's really cool. You get double the bang for your buck. You do your physiology, and then you go and look in the tissue. And they have increased photostability, but the Alexas are smaller. So one of the things that immediately becomes apparent is that there's steric hindrance issues with these guys because they're nanometers in size as compared to an organic fluorophore. And unless you have a partner, it costs a ridiculous amount of money because Quantum Dot Corporation has a patent and they charge you a lot. So getting a partner was really important, and it turned out there was something called the Center for Integrated Nanotechnologies. I put in an application for the user jumpstart, and I just came from a meeting in the Los Alamos lab, so I now have a partner, and she's going to make lots of different dots for us. And we're going to work together to develop biocompatible dots. I'm very excited. So we'll be spending time at Los Alamos labs. And I'll skip that slide. And so we're very interested in using these for optical imaging and looking at the actin cytoskeleton. People are also injecting them into vessels and looking at blood circulation. We have concerns about toxicity and steric hindrance, so you use them, but you also wonder, okay, how do they perturb the system? And so we started looking at them in cultured cells, and this just shows you a fluorescent picture. These are what the particles look like. When you look at them, this is where they are. These are targeted quantum dots being taken up by cells, this is the no Q dot image. These are, and this also, the cells were imaged using a cell tracker red, which lets you know the tissue is viable. And so we've taken this to TEM. And when you go to TEM, you can actually see that the particles are internalized. So you actually get to see where things were later. So it's really a neat technique. So we're working now with the Center for Integrated Nanotechnology. So now we have a national lab collaboration. And so this is kind of where these things are. And so the future directions for the research is to continue what we're doing with the genetic and the anatomy. We've invested in that. But really to start moving to an organ culture model for regeneration, to start using systems approaches in computation. We're going to need to collaborate with more mathematicians. And then to collaborate with our SINT investigators and with our colleagues at Southwest Sciences. And I need to acknowledge the wonderful students who over the years have worked in my lab. They're a terrific group. I'm very proud of all of them. And of course, our funding agencies. This work is funded by NIH. I've had seed money from NSF, NASA, Whitehall. I'll write a grant to anybody, and no grant is too small, nor too big, you know. And so, and then of course, these are all the student programs that have supported students in my lab, and these are some of our collaborators. And of course, I never ever forget to acknowledge the Xenopus who have been drafted into this research, but I am grateful to them for their participation. And so, thank you. So, I know that was a lot, but
Questions?
if you have questions.
You know, in your organ culture model, are the hair cells electrophysiologically still active?
That's,
You tested that?
no, and that is obvious.
Is
It's
that
not to
going to
happen.
be easy to do?
No, it's not going to be easy to do. It's very hard to, the way people work on these is they do, they dissociate the cells. And so we would have to do electrophysiological dissociations. The other way to look at that is to try using voltage-sensitive dyes to see if they can be stimulated. But that's, that's the difficult part of working on the ear, is that the electrophysiology. I've done actually in my early days electrophysiological work dissociating. But when you start, and I was really excited about that, but then when you start looking at the anatomy, you go, what more am I going to learn from patching a cell out of the whole tissue? And so we've tried to patch in whole ear. It turns out that the ear, because I think if you look at the ear, it has so much liquid in it, and I've thought about this, it has a really tough protective sheath around it. So when you try to do, for example, a slice prep, and you try to go through, and the people that we were collaborating with were just astonished, the electrode is like rubber. You can't, it just bounces off. So great question, obviously an important question. Don't worry.
Well,
Yeah.
do you know is the origin of the ear then mineral crest derived?
Yes, it is. It is epithelial, neuroectodermal tissue.
So they understand the developmental events that lead to it pretty well, or? is it
No.
all-
Okay, so Andres Collazo, so I have like a counterpart person, that's Andres Collazo. So he works, I pick up like at stage 37. So the Xenopus people have worked on the early stages. So Andres tackled that. He did a lot of injections of dye, and what he found, and actually what we found also from the VRBU at a certain stage, is that the ear forms by a lot of migration and mixing. And it's probably true of a lot of tissues, but it's particularly true of the ear. So early on, you have the tissue, it's dividing, and then at a certain stage, about the 40s, you start getting tissue reorganizing and moving around and stuff moving in, and so it's a complicated tissue in that regard. And when you think about it, at the end, you've got blood vessels in there, you've got bone around it, and what's the ear? Is the ear only what's inside? And the ear itself is going to have, all those nerves are also vascularized. When you do a surgery, you have blood vessels that come right to the very end, and huge mitochondria, which you see in Xenopus, providing energy to them. So so wind, people are really, the wind and the BMP, those are still important. And I'm really intrigued about, asking the question, well, I wonder if there's local issues with wind involved BMP later on in development. But yes, there's interest in the wind signaling pathways and the BMP, bone morphogenetic protein pathways.
So with the quantum dots,
Mhm.
is there a limit to the number of different colors you can make?
No, provided you have the filters that can separate them. Okay?
Because the reason I was thinking that is I know they do a lot of that with the bioinformatics, the co-expression mapping,
Yeah.
where they take data from all
Yep.
different lives and
Yep.
then look for
Yep.
things that are always
Yeah.
expressed at the
Yeah.
same time. So it'd be interesting to take that information and then to actually make predictive models.
Yes.
And then have all
Yeah.
the little quantum dots that actually show that, yeah, all those receptors are on the same
That's a
side.
great idea, Lindy. That's a great idea. And I think that's exactly the kinds of ways people would like to use these. And so, yeah, because they're the same material. But the issue, and what you learn when you start getting into this, is that the fabrication isn't trivial. Because then you have to be able to say to the person, and okay, if they're really tunable, and I saw this right away, so now I'm going to show you the nitty gritty. And this is, so here, they would always show you this picture when you get the picture of the quantum dot. And they would go, oh, multiplex, and we tune them by the size. And I'm looking at this and going, does not compute, does not compute. And then I realized what it was that was disturbing. First of all, these are pretty wide. If you're telling me they're tunable, I think that's a lot of noise. But I think in part, that's the vendor gives you that partly reflects that I think you're getting a distribution of dots. Right? It really has to be, they're physics. So this means that that's the best quality control they can do when you buy that. But looking at spectrum going, something's wrong, you go, oh my God. And this is what they don't show you. See, this is bigger here, right? So if you hit these guys, if you normalize these for the same wavelength of light, the bigger guy, this guy gives you, is brighter. And so you comment, wait a minute, the red ones are brighter. And you go, yeah, they are, because they're bigger. They're going to absorb more for the same amount of light coming in. So then, if you do that, then you start, these guys should appear like this, these guys should appear like this, and then the overlap in the first one. And when you call them on this, they go, yes, actually, you have to use different filter sets to do a quantitation. Do you understand what I'm saying? That these really should be bigger because this is bigger, this is really big. Anyway, so yes, there's a lot of interest in these. For me, it's fun because it's working. For me, it was fun to go to the synth meeting because you walk into a room And these are the people that are making the stuff that's going to change our lives. These are engineers. These are people that are really, the things that I saw them design, they're making cantilevers that can measure atoms, atomic weight cantilevers. They're making biomems devices so that you have tiny chips that you can do cell sorting with. Really, and I feel like I would like more of the people I know to know this is happening, because it's idea driven. And so they'll only do what the engineers can think of and any biologist they talk to, but I walk away from those and start having ideas about things I did not think were possible. Then the other part of this is just practical. If you go to NSF, the budget's all in nanoscience. That's where the budget is. And nanobiology is a huge deal, and cancer, nanobioscience, using these dots to functionalize them, target them to cells, and use them for treatments are a big deal. Now, the other side of that, there's interest in ecosystem flow. If these things get in the environment, where do they end up? I've actually thought of plant projects. Arabidopsis, actually, we can put little Arabidopsis in the Q dots and see, are they taken up? So there's a concern at this meeting that the nanotechnology could go the way of GMOs, where people are going to go, oh, bad scientists, kind of doing negative things for the environment.
Check the lung tissue of your students.
Pardon me?
Just check the lung tissue of your students.
Exactly, yeah.
I was saying, I was telling
see.
her before, like the Berkeley Nanotech Lab, Berkeley constantly is trying to shut it down because
Yeah.
they don't, I mean, they haven't really tested.
Yeah, and so that's actually the angle that Jennifer Hollingsworth and I are doing. She has actually a novel material that she's working with. So she's carved out on her end a particular material. I said, because right away I said, if you're going to do long-term studies, I need to know they're not toxic. Otherwise, I'm studying the toxicity of the nanocrystal. So we right away went that angle, and now you're seeing lots of calls for biocompatibility. So that's what she and I will be doing. But I'm really excited because these as tags, I'm not so sure they're going to be so great inside cells because they're big. But I think as probes on the outsides for receptors, if you have a ligand, they could be great. And they really are bright. They really, really are much brighter. What people are doing is because they heat up, because they're a material, they do heat up. They don't fluoresce, so that energy has to go somewhere. And I think there's a proof of concept of this already, and this is cool, that the tissue gets loaded with it, and when you shine the light on them, and you can use blue light on these, they heat up and it destroys the cell. So if you're a cancer cell, or a tumor, that's a way to destroy a tumor. And then I go, okay, and then where do all those nanocrystals go? And so who are you anyway? Why are you asking that? And so then the biocompatibility issue
Do
comes
they
up
know if
again.
they're magnetic or anything at all? 'Cause
Oh, you can make them. See, this is the fun part about talking to you. Oh, yeah, you want a magnetic? Yeah, I can add a magnet. Because I asked her that. And so my collaborator says, yeah, we can magnetize them. Now, what's interesting is that this is Los Alamos Lab. So what wavelength is our government really interested in? Far, far IR. Far IR. Because I was telling her, if we do this, I don't have the microscopes, right? Because we don't have the microscopes. Remember for UV technology, they had to recode the objectives. Then we went to multiphoton. Everything's material science. So the lenses were all coated with things that did not pass the IR. People were like, can't see my MP signal. Of course you can't. The coat on the lens won't let you see it. So it had to develop new technology. And so they have microscopes there that are configured for that. So the far IR technology, the government's very interested in as a report. And you can imagine that we could all be dusted with nanoparticles that were in the far red, but we even know. But if you have them, where have they been and where are they going? You would think that would be strange, except that I talked to the robotics and insects people, and there are actually people putting little cameras on insects so they can fly over and do reconnaissance. And it's like, okay. So I don't think like that, but obviously people are thinking that way and they're doing this work.
The other thing I was going to ask you was, what do you think about the idea that the centriole itself is a determinant of directional location?
I am so interested in that concept. And so here's what I didn't show you. So Marty got a cetylated tubulin. Originally we used that because that's a neural marker of stable neurals, and we used it in Germany. And what we found is that the hair cell eventually expresses this. Well, of course, it's a neuronal-like cell. and that the kinocilium does. So we started looking very early in development, and we've seen this, I'm going, buddy, it's green everywhere. And then it's just like just background, and then we didn't see it in the other one. And then we look in our images now, lots of the cells have a tiny, tiny little, like a little bud that's kinociliar. And I think the literature is showing that it's, again, mechanosensor as being something fundamental to biological processes. So yes, I think that that is something that definitely this is a system, and there are people in my field doing it. And so what we want to do now is go back and look at our SEMs more carefully to see what that kinocilium is doing, or even with the labeling of the kinocilium, trying to do some kind of modeling. But yes, directionality.
One question
It's
for-
just one
Yeah,
thing.
I'm sorry, I'm
So how
sorry.
important are the SEM and the multiphoton confocal to your work here?
Well, let's just say that I survived when my lab burned. So, SEM less so, although I think from material science perspective there could be interesting things. Confocal, extremely expensive. Yes, expensive. Very important, but you know, I'm looking now that a lot of the spinning discs for some of the stuff we're looking at would be fine. We're really moving to the organogenesis and the array and the gene expression. And you know what? I have so many friends now, I'll just take a trip and put my stuff on another confocal. Yeah.
But we have the confocal here.
I know I wanted to see that at some point. I know we're meeting. I'd like to know more about it.
Okay, great, okay. thank
I'm
you.
so sorry I ran over, but you